Hi everyone, and welcome to the last of our lab lessons. Try not to let the tears get onto your laptop or your computer that you're using right now. I know you're going to be so sad without this class, but for this final lab, what we're going to do is we're going to just go through a few other examples of biotechnology that we may not have covered throughout the course so far. Now, in addition to plasmid, sometimes we use some other types of vectors. Here we have bacteriophage lambda vectors. That's basically saying that you can use lambda, which is a virus, okay? A virus that infects bacteria with its viral genes. And interestingly, the viral genome of lambda is not actually all essential. So there's a portion of it that you're able to just, like you see in this figure, you're able to just cut out part of that genome and glue in whatever genes you want and put it back into the bacteriophage, into the virus. And now that virus will go and inject that DNA, including the gene you added, into bacteria. Okay, so this is just an alternate route that sometimes researchers use. And when you do this, when you have a viral vector introducing DNA into bacteria, we call that transduction. Okay, so in lab you perform transformation. This here is transduction. Now, another type of research vector that is commonly used is what we call the TI plasmid. This one's used for plant engineering. TI stands for tumor inducing, okay, not a famous musician. So it is tumor inducing, and the reason it's called that is because it's actually naturally carried by what's called Agrobacterium tumefaciens. And you have all probably seen this at one point or another. That's actually a type of bacteria that gets into plants and especially trees through cuts in their, in their bark. So we call that wounds. So for instance, if your parents ever yelled at you, if you're, basically carving into a tree or picking away, peeling with your finger pieces of the bark, when you wound that tree, you open it up to this bacteria. And that bacteria carries a plasmid that has what we call tDNA. And the tDNA, or tumor DNA, basically what that does is what it says, it's tumor causing. It integrates into the plant genome. And what that tDNA has is auxin genes, cytokinin genes, and opine genes. And these are genes that produce enzymes that, as you can see from down here in the figure, promote cell growth, cell division, and feed that bacteria. Now, when you think about this bacteria is putting genes that give enzymes to produce more cells, more plant cell division. What is that? That's cancer, right? That is plant cancer. And that's when you see those giant bulbous tumors sticking out of trees that you're going to see on the next slide. So anytime you're walking by or driving by any of the trees, even on campus, that have these big growths sticking out of them, I want you to shout Agrobacterium tumefaciens, okay? Just shout at it. Look at it. You see, look at that tumefaciens. And all your friends will, of course, think you are the coolest person ever because, that is awesomely cool, of course. Okay? Now, we can then use these plasmids in genetic engineering because if this bacteria has the plasmid, which mediates transfer of DNA segments into plant cells, well, then what we can do is we can cut out those genes for the tumor induction and instead put our own genes in there. And the rest of that plasmid is very good at integrating into plant genomes. Okay, so we're able to modify that plasmid, the TI plasmid, to take out the bad bacterial genes and put in our desired genes, and boom, now we can get the plants to produce whatever we want. Okay, so to help you visualize what I was just talking about a minute ago, this is that, crown gall tumors that you might see on campus or various places when you're walking around or driving around. So remember to shout, look at that tumor fascians, okay? And this up here demonstrates what we were just talking about in terms of using the TI plasmid for genetic engineering. So you're able to cut it open, remove those tumor-inducing genes, and ligate in the genes of your desire. Then when you introduce this plasmid into plant cells, well, that plasmid, as you can see here, is naturally very good at integrating into integrating into plant cells and into plant DNA. So now the plants produced from that cell will have your desired genes. So for instance, those genes, think about what might you want to give to a plant. What kind of traits? Well, maybe it's a gene to produce bigger fruit. Maybe it's a gene to produce more fruit, more vegetables, okay, more product. Maybe it's a gene to grow taller, grow bigger. Okay? Any of these things resist pests, such as, bugs that might try and attack those plants. So that's how we do genetic engineering a lot of times when it comes to plants and crops, is we use the TI plasmid. Okay? Make sure you know, if you see a crown gall tumor, though, that's thanks to Agrobacterium tumor fascians. Okay? Remember that. It may pop up again. Okay, so when we talk about genetic engineering, we also have what's called gene libraries and DNA libraries. Now, basically what this means is not only are we capable of cloning single or individual genes, but now we can clone libraries of whole genomes, okay? So as you see in this figure here, we can take an organism, let's say a bacteria's entire genome, so every gene it has, and what we do is we chop it up, and each piece ends up in a plasmid, and then we do transformation, and you get a single plate that has all of the genes of that bacteria's genome represented in that single plate. Okay, now keep in mind, each colony will not have every single gene. Okay, it's the whole plate that's the library. Okay, the plate will have all different bacteria, and each different colony will have different genes. So let's say this guy here has this first part. This guy here will have this second part. Okay, so each bacteria on that plate will have different genes, but the plate represents the whole genome. And since that plate represents the whole genome, you can then screen the plate to pick out whichever genes you want to work with. Now, there are also with gene libraries what we call metagenomic libraries. So they'll have a plate that has genes from many different bacteria. So a lot of times you see this, for instance, from an ocean environment. They'll collect a sample, and then they'll have a plate that represents all the genes in that particular location. They have prokaryotic gene libraries, which are, for instance, bacterial. They also have eukaryotic libraries. So these will have a lot of times yeast genes, or even you can do this with whichever organism you want. But basically the premise is the same, that the final plate will have the whole library represented. Then that brings you to the next point. Once you have this plate with all of the genes of an organism represented, well, what do you do with it? You have to screen it, right? Because now you want to know particular genes. Are certain genes there? Okay, where exactly in that plate are certain genes? So there are two techniques that we can use for screening a gene library. You can use hybridization, or you could use immunological screening. For hybridization, I want you to write down that this is for DNA. So you're using DNA probes, you're using single-stranded complements that are nice and sticky, and you're screening the library for the DNA, the actual genes. Immunological, you're now using antibodies to screen for gene products, okay, the proteins produced by the genes. Okay, so hybridization, you're going to be looking for DNA. Immunological, you'll look for proteins, particular proteins. Okay, so the first method is hybridization screening. Okay, I'm going to go through first, and then I'll give you the recap slide for you to take down your notes. So the first thing that you would do with hybridization screening is you would transfer your bacteria, which contain the fragments of the library, onto a filter. Okay, so transfer all of these bacteria from your plate that have all the genes present, and you transfer them onto a thin filter. You then lyse these bacteria, and you denature the DNA with alkaline or very basic solution. Okay? So now when you have denatured DNA, what do you know about single-stranded DNA? Well, single-stranded DNA will like to try and find its partner. Okay, it will want to stick to its partner. Okay, so what you do then, once you have this membrane of all the single-stranded DNA of this library, you then cover that filter with a solution that has radioactively labeled single-stranded DNA probes that complement the exact gene that you're looking for. Okay, you then develop that, and wherever you see the black, or in this case blue spots, that tells you that that's where your target gene is. Okay, that's how you find your target gene. So please remember that for hybridization screening, the key here is you take all of the bacteria of the library, you transfer it onto a membrane or filter, you break open those bacteria so it's just the DNA that you denature to make single-stranded, and then you target it with highly specific single-stranded probes that you can then detect. And those single-stranded probes will only bind to the target gene that you're looking for in that big library. Okay, so I'm going to give you a recap slide so that you can take down notes. Okay, so here you can pause the recording and write down the notes. The other way that you can screen a gene library is immunologically, meaning you use antibodies. So now it's the same kind of concept, but now looking for gene products, looking for particular proteins, because not only do you want to know the bacteria has the plasmid, but you want to know that it's functional. Okay, that you know which ones are actually producing the protein of interest. So what you do is, just like with the other one, you take those colonies from the plate, okay, and this time you're transferring to a membrane, and you're lysing them just like in the hybridization. So now you break open all the bacteria from the gene library, but now it's the proteins that you're targeting. So you use a different filter, a very particular membrane filter that the proteins will stick to. And the specific protein that you're looking for, you can then target with a primary antibody, because primary antibodies are specific to exact proteins that you would look for. You then apply the more general secondary antibody, which will bind to any protein, right? Any antibody. And that secondary antibody, what's important about that one is that it has detection on it. Okay, so it'll have some sort of probe or detection system on it. Usually we use chemiluminescence. So basically you want to see fluorescence. It's kind of like thinking in terms of a Western blot where you get the fluorescent detection. So kind of think of this like screening for a particular protein, where it's almost like hybridization, but now instead of DNA, you're looking for a protein product. You take the gene library, you crack open all of those bacteria onto a membrane where their proteins will stick, and then you look for the exact protein that you want. And keep in mind through this, whether it's hybridization or immunological, you still have the original plate of bacteria. It's kind of like you made a copy of it, right? So once you find the location with the fluorescent detection, you know exactly which colony to go grab and allow to grow. Now, when we talk about PCR, there's a lot of different types of PCR that, basically they have made this technique more and more powerful for us researchers. One of the ones that we use a lot is called qPCR, or quantitative real-time PCR with TaqMan probes. Okay, what this means is not only can we amplify or make a ton of copies of the DNA, but we can quantify it as well. So this one will quantify while it amplifies because it's using these special TaqMan probes. And what these TaqMan probes are, you have to know what they are. They have a fluorophore on one side, so a fluorescent probe on one side, and then what's called a quencher on the other side. A quencher, like when you quench your thirst, it blocks the fluorophore, okay, because right now it's connected to that guy. So when the quencher is near the fluorophore on this TaqMan probe, the quencher blocks fluorescence. What then happens is this TaqMan probe, it's a single-stranded piece of DNA that binds in the PCR to the target DNA that's getting amplified. What happens when the polymerase is amplifying that DNA? Taq polymerases have what's called exonuclease abilities. So that Taq polymerase, as it's building the new strand, it will then come along. Once it makes it to this probe, it'll chop it up. So when the Taq polymerase here chops up that probe as it's building the copy of DNA, what happens? The fluorophore is now floating away. It's separate from the quencher. What happens when the fluorophore is separate from the quencher? It fluoresces. Okay? So the more initial product that you have, the more template DNA you have, the more new strands will be built, the more probes will get cleaved, and the more signal you'll see. So that allows us to quantify it. Okay, so one more time. The more product you have, the more template DNA you have, the more new strands will be built with the Taq polymerase, which means more probes get cleaved, which means greater fluorescence as the fluorophore is separated from the quencher. Okay, this is a slide that helps you kind of see in words what I was just saying. Make sure you're very comfortable with knowing what happens with the fluorophore when it's near the quencher versus when it's separate. Okay, make sure you can explain how exactly does a TaqMan probe allow us to quantify a PCR product. Okay. Similar to that, sometimes you will hear reverse transcriptase PCR. what reverse transcriptase PCR is now you're amplifying genes using mRNA as the starting point, because as you know, reverse transcriptase means you're going from RNA to DNA. Now, why would you want to use mRNA as a starting point of your PCR? Well, this is used in gene expression research, okay, because mRNA is gene product, so it shows how active particular genes are, okay? So you're now going to have to clone copies of intron-free DNA in this process and amplify that. So the main steps of this, of a reverse transcriptase PCR, first you have to isolate that RNA, the mRNA. You then clean it up to get rid of any DNA that might be contaminating it. And then you do what's called first-strand cDNA synthesis, and you do not have to memorize these steps of first-strand synthesis. It's basically meaning that the machine will make the complementary DNA copy of that RNA, okay? And then once you have the DNA copy of that RNA, now you can put it into the PCR and amplify it. Okay, because PCR only works for DNA. PCR can only amplify DNA. So if you want to study RNA with PCR, you first have to make a DNA copy. So that's why you need reverse transcriptase PCR. Okay, keep that in mind, circle, write down that note, put little stars next to it. PCR only amplifies DNA. Okay? Now that reverse transcriptase PCR is very valuable to us in the research field because it allows us, like I said, to examine gene expression. Okay, so you can look at whether a gene's expression is upregulated or downregulated in certain conditions. Basically meaning you'll have two different conditions and you'll ask yourself, when do I see a greater signal? When do I have more of that fluorescent final product? So if you see a lot of reverse transcriptase PCR product, well, that means that there was a lot of mRNA present to turn into the DNA and make copies of it, right? So that means that gene expression was high in that condition. Whereas if you do not see much fluorescence or amplification at the end of the reverse transcriptase PCR, well, that tells you there wasn't DNA to copy or amplify because there wasn't mRNA to copy, which means the gene was not being expressed. Okay, so reverse transcriptase PCR lets us look at how much gene expression there is in different conditions. Okay, so it's a very powerful technique. There are also a lot of medical applications of PCR, whether you're doing regular PCR or the big time qPCRs, okay? And with these, bullet points that I have on this slide, keep in mind that there's a whole lot more you can do with PCR. This is just the sampling. It's very powerful technique because you're able to amplify or make copies of very small amounts of DNA, which if you think about it, well, that's great in forensics, especially since at a lot of crime scenes, you're not getting a lot of DNA or it's been highly contaminated. So this allows you to kind of amplify your sample to work with. It's also very powerful in diagnostics because we know the genomes of almost every bacteria virus that are most commonly infecting people. So whenever we find new viruses, new bacteria, the first thing we want to try and do is, get its genome, sequence its genome. And then once you can do that, you can use PCR to see does a patient have it or not. For instance, you use primers that target sequences found in that virus or that bacteria, and you test your patient's blood samples or swabs or pus samples, anything like that, just to see, okay, if this person's infected, the PCR primers will pick up the viral or pathogen genes and make copies of it. And you'll see those copies on a DNA gel, like on the bottom of this slide here. Okay, that's DNA gel. You can see bands when the amplification occurred. Now, if that patient's not infected, the primers won't find anything. They won't have anything to bind to. There won't be any amplification, and you won't see any band on the final gel. Okay, and they do this for a lot of different infections, and they even look at, mutated genes like BRCA1 for breast cancer. They'll have primers that target the mutated sequence and see if that's present in a sample. If you see amplification, that means the gene you're looking for is present in that person. And if you don't see amplification, then the primers had nothing to stick to. There was no bad gene present. Okay, so very powerful in the medical field. It's also very powerful in the environmental field. Okay, environmental field, I'm not going to go into too much detail here, but I basically wrote out a lot on this slide just in case you're more curious about how it's actually used environmentally. But basically, you're able to target bacterial genes to see if they're contaminating certain areas, or to find particular metabolic aspects that might be valuable from different bacteria. So for instance, if there's a hot spring, these very crazy conditions that can withstand heat, well, let's gather the microbes there and amplify their genes, see if there's a product that we can use. Okay, so we're able to find very valuable enzymes for our research, even things like the PCR polymerases that we use. We have gotten from, trawling ecological samples, looking for amplified gene sequences. And then the last one here that we have is the idea of rescuing DNA from extinct life forms with PCR. Now, this is kind of like the Jurassic Park type of scenario. It's not completely, realistic in some senses, because cloning such large amount of genomes can get problematic. A lot of times the vectors can't handle it, or you have a lot of mutation issues or instability issues. But just be aware that trying to rescue old forms of DNA is one other possible use of PCR, because basically once you look at sequences, you have sequences. If you can make little primers for them, you could amplify or target any particular genes you want. Okay, so we're not going to go into crazy detail. I just want you to know that there are environmental and extinct life form uses for PCR. And that is it. You have made it through the final lesson. It has been an absolute pleasure teaching you this semester. You've all made me so proud of the kinds of answers and the questions that you pose in the Remind app and throughout the assignments that we had in this class. As you prepare for the final, please contact me with any questions that you have, and in all of your future semesters, if you ever need anything, whether it is help with understanding anything from your other classes or advising regarding future careers, jobs, and graduate programs, never hesitate to send me an email. Thank you and have a wonderful day.